In asthma, like in sinonasal disease, the inflammatory cascade begins at the airway epithelium. Allergens, microbes, and pollutants trigger the release of epithelial cytokines or alarmins. Similar to the upper airway, these epithelial alarmins initiate the release of downstream cytokines from cells of both the innate and adaptive immune systems. In asthma, there's a similar heterogeneity characterized by cytokines in cells of type 1, type 2, and type 3 inflammation. As in the upper airway, type 2 inflammation is characterized by much of the same cytokines and cell types. Additionally, eosinophils are associated with increased basement membrane thickness, mucus plug formation, and perpetuating T2 inflammation. Eosinophils release granular proteins, which by interaction with DNA and histones form eosinophil extracellular traps, or EETs. EETs are intended for protective mechanisms against pathogens. However, left unregulated, EETs enhance type 2 inflammation, mucus plugging, stickiness of secretions, and epithelial disruption. We continue to learn about the role of EETs in health and disease of the airways. Th1 cell activation leads to the release of interferon gamma and TNF alpha. Th17 cell activation leads to the release of cytokines including IL-17, IL-22, and TNF alpha. Several of these cytokines contribute to alterations and hyperreactivity of airway smooth muscle or ASM cells and airway remodeling. Recruitment of macrophages and neutrophils results from activation of both Th1 and Th17 cells, leading to type 1 and type 3 inflammation respectively. Among other functions, airway neutrophils release proteases and nuclear contents that result in generation of neutrophil extracellular traps, or NETs, and activate the NOD-like receptor protein 3, NLRP3 inflammasome. NETs and inflammasomes result in lung parenchymal tissue destruction, increased mucus production, persistent inflammation, and lung injury. In COPD, oxidative stress, microbes, and pollutants such as cigarette smoke trigger type 3 innate lymphoid cells, or ILC3, and Th17, and thereby release of cytokines IL-17 and IL-23, as seen in asthma. These cytokines result in neutrophil-mediated inflammasomes and proteases, and type 3 inflammation, important in parenchymal destruction. In response to these stressors, the epithelium also promotes the release of IL-33 and TSLP and amplification of type 1 inflammation. As seen in the upper airway and in eosinophilic asthma, a subset of COPD is also characterized by type 2 inflammation and eosinophilia. Compared with the healthy airway, the asthmatic airway demonstrates disruption of the epithelium, goblet cell metaplasia accompanied by an increase in mucus or mucus plugs, thickened ASM, thickened subbasement layers, and airway fibrosis and remodeling mediated by a single or multiple types of inflammatory cascades. Similarly, COPD can manifest airway epithelial and subepithelial changes due to type 1 and type 2 inflammation. Emphysematous COPD additionally is accompanied by loss of elasticity and parenchymal destruction due to type 3 inflammation.